Okay, so thanks.
Relay this.
Yeah, thanks, Jean-Luc, for that nice introduction, and thank you very much to everyone that's been hosting me here. I've had an excellent time, and it's a beautiful campus to walk around as well. Thank you. So the topic for today is genomic selection for more efficient food production. Just a quick outline of what I'm going to cover. First of all, what is genomic selection and how can it help with this challenge of producing food more efficiently? A recent success story that I'd like to tell you about that we've just had in the past couple of months. And then where we're taking the technology in the future. And there's two stories there. One is about incorporating whole genome sequence data into our genomic predictions of breeding value or future phenotypes. The other is a little bit different, but it's taking the genomic selection methodology and applying it to predictions from the microbiome that you find around animals and around plants and so on to predict future phenotypes. So animal and plant breeding, as you're well aware, are very powerful sciences, and they've really contributed spectacularly to this challenge of producing more food and producing it more efficiently. So in animal breeding, for example, the time to market weight for meat chickens has decreased from 16 weeks to 5 weeks in 30 years. So that's a pretty spectacular decrease. In corn breeding, you can see the yields in hybrid corn have just been increasing year after year. So it's really contributed a lot to this efficient food production. The problem is we now need to go quite a bit faster in order to meet this challenge of many more mouths to feed. So how are we going to do that? One opportunity we have that we didn't have, say, 5 years ago is we can now get an abundance of DNA information really cheaply. So those of you that have been working with DNA for the past decade or even longer, you'll know that the cost of getting a DNA marker, one bit of information, 1 point, has gone down by 1,000 times or more over that period. So it's now possible for just about any species to get an abundance of DNA information. And this is true of cattle. It's also true for the different plant species through the nice work that's being done at Cornell here on approaches like genotyping by sequencing. It's just driving that cost right down. The challenge then is to take advantage of this new opportunity. How are we going to do that? And this is even more challenging because of the genetic architecture of the traits that we're mainly interested in. So this certainly isn't true for all traits that are important to us, but for many of the traits, they have a genetic architecture where there's many loci, all of small effect, that are contributing our genetic variation. So it's absolutely not the case that you can find the gene for yield and then increase the frequency of that to increase yields. There are many genes contributing to yield. Same with milk production in dairy cattle, many genes contributing to that genetic variation. And this is just a result from the classic Illinois corn experiment where they selected for divergent oil content. And when they recently dissected the genetic architecture of that trait, how many different genes were underlying the differences they saw that they needed at least 50 genes to explain 50% of the variance in that trait, and that's only 50%. To capture the remainder, they need a huge number of extra genes. So 2 challenges: how can we use this DNA marker technology, and how can we use it to deal with traits like this where you have many loci of small effect contributing the variance? So the answer, I think, is genomic selection. So the starting point for genomic selection, what you need to get it going, is a really good reference population or a training population. A large training population where you have many individuals, be they plants or animals, that you have measured for your target trait, say it's yield or disease resistance or milk production in cattle, and you also have genome-wide DNA markers on those individuals. So DNA markers right across the genome. So this is just a little cartoon of that. This is a chromosome. You've got the chromosome here, you've got the markers, and then you've got the genes affecting that trait. So the next step is to do an analysis and to see how much of the trait differences those markers explain. So for this particular marker, it's a SNP, single nucleotide polymorphism, 2 alleles. If you get a copy of the T allele, you get an extra 2 grams of yield. And that's because it's tracking a gene right beside that marker that increases yield. And the trick with genomic selection is not to do this one at a time, but to fit all those markers simultaneously. So if you've got 50,000 markers, you estimate the effects of all the markers on the trait simultaneously. And that gives you a prediction equation. And the prediction equation is just the effect of the second allele at every marker on your trait of interest. So the first marker, it might be 2 grams of yield, second one might be 0.2, and so on. And that thing, if you've got 50,000 markers, that's going to be 50,000 long. Once you have that, you're really in business, because what it means is you can go to a group of selection candidates, for example, young plants that have never been measured for yield, or young dairy cows that have never produced any milk, and you can predict those phenotypes. You can genotype those individuals for the 50,000 markers, run their genotypes through the prediction equation, and then get estimates of their breeding value. What's the genetic merit of those individuals? And then you can make selection decisions on which ones have the best genetic merit, select them, and turn over your generations. Now, if you've done a really good job with your reference population, if it's really big and you've got lots of markers, you can do multiple cycles of this and make extra genetic gains by doing that. So that's the theory behind genomic selection. Next, I'd like to take you through an example where it seems to be working pretty well. And that example is in the dairy industry. So in our reference population in Australia for, this is Holstein Friesian, black and white dairy cattle, we now have 3,800 dairy bulls and about 10,000 cows in our reference population. And they're all genotyped for 56,000 genome-wide markers. So we can use that reference population to work out the effects of each of those markers on something like milk yield. And the phenotypes for the cows, it's just how much milk they produce. For the bulls, it's the average of their daughters' milk production. So each bull might have 100 daughters. I'm not sure if I'm going to get this right, but a plant example might be the cows are a variety that you just have a small number of replicates on, not a very accurate phenotype, and the bulls are like a variety you have an enormous number of replicates on, so an extremely accurate indication of their genetic merit. And we combine both those sources in our reference. So we've been running genomic predictions off this reference set for the past 2 years, I think, or 3 years. And we've been giving these out to the dairy industry. So to bull breeding companies, we've been saying to them, this is the genetic merit of your bull. You can go and sell these bulls on the basis of this to dairy farmers around the country. Okay, so, and the companies have believed us and have been taking this information and using it. Now, the first bulls selected on these, this technology's been commercialised, but the commercial breeding values, they've just got their first daughters that are milking. So what we can do now is compare what we gave to the companies, what we said the genetic merit of the bulls should be, to what it actually is, based on the performance of their daughters. And this was one of those moments in your career, my career, where you're shaking as you do the analysis because you really want to know the answer, but if the answer's bad, you might have to retire very early. But luckily, it wasn't too bad. So what do we have here? Along the x-axis, we have the breeding value that we predicted just from the DNA information, how good those bulls would be. And then on the y-axis, we have the information after they've got daughters. And I'll split it up here. The more daughters the bulls get, the more accurate it is. So you can see the way it changes as they go up to 70 daughters. But the result was we did a pretty good job of predicting which were going to be the really good bulls and which would be the poor bulls, which was excellent. And the amount of movement around the line, we certainly didn't predict absolutely the best bull in every case, and we didn't predict it bang on the line, but we did tell the breeders there would be a certain amount of movement related to the accuracy of these breeding values, and the amount of movement we saw was pretty bang on the accuracy. So that was great. Just how the dairy industry is actually using this to increase genetic gain. When you use this technology of genomic selection, you've got to look for the opportunities where you can actually increase genetic gain. And the opportunity in the dairy industry is through turning over multiple generations in the time it previously took you to turn over one. So the way the programs used to work is you would take a whole bunch of bull calves, maybe 100 of them, selected on how good their parents were, but that information was nowhere near good enough to sell out to the industry. It's like having a variety in just one plot, I guess, that you've ever evaluated. So what they would then do is progeny test those bull calves. So mate them to cows, have daughters milking, and eventually get very accurate measures of genetic merit on each of those bulls and then select them. What's happening now is the genomic breeding values are accurate enough to go into a population, screen out the ones that are going to be very good, and just pick out the top to market out into the industry. And these bulls might only be 18 months old. And what you've done there is really reduce down the generation interval, the time it takes you to turn over a generation interval. So now you can actually turn over 2 or 3 generations in the space it took you to turn over one. And this is actually happening. People are trying to go faster and faster at the moment, trying to squeeze 3 generations into there. And your formula for genetic gain, where you've got the intensity of selection times the accuracy times genetic variance divided by the length of generation, if you cut your generation interval in 2, you go twice as fast. One thing to point out though is the genomic breeding values at the moment, even for dairy, aren't as good as a progeny test. They're still not quite as accurate. So you take a bit of a hit here, but you more than make up for it in the reduction in generation interval. Okay, so that's the good part of the story, but we know, and I think it's useful to point out, there are limitations with this technology. Even in dairy where it's working really well, we are seeing limitations. The first one is the accuracy is lower than a progeny test. So we have to convince the breeders to use something that's less accurate than what they had before. They can just spin it faster though. For some traits, fertility for example, disease resistance, it's quite a lot lower accuracy than for something like milk production. And one problem we're really running into is the accuracy decays over generations. In theory, with genomic selection, what you could do is just keep spinning the generation, selecting only on genomic breeding value once you've estimated the marker effects. What we see though is once you go 1 or 2 generations away from your reference population, the accuracy is really starting to drop off. So your predictions of genetic merit have a very large error around them. And that's in a way a shame because we'd like to spin the generations faster, but the technology at the moment just isn't up to doing that. So 2 generations probably at the most. So the question is, how are we going to fix this problem? How are we going to take it to the next step? And that's where we come to sequence data in genomic predictions. So before I get into that, there's 2 ways of increasing the accuracy of these genomic predictions. You want the accuracy to be as high as possible, so you get the most genetic gain. One way that we've seen tried and tested is just to increase the size of the reference population. So more and more individuals that you have phenotyped and genotyped to estimate these marker effects. And this is number of bulls in the reference, and this is accuracy here. We have a theoretical prediction based on genetic diversity, size of reference, and heritability of the trait. And this is data from the US, actually, where they increase the size of their reference population over time and the increase in accuracy you get. And you can just see it goes up and up. But it does get harder and harder to make improvements in accuracy. You need more and more bulls or more individuals to make the same improvement. The other problem is we know now that this line, this curve, has an upper limit as well. It actually plateaus in an upper limit. And that upper limit is the proportion of the variance that your markers explain. Even with 50,000 SNPs across the genome, that's still not good enough to track all the genes explaining variations in things like fertility and milk yield, for example. For milk yield, it goes pretty close, but for traits that have been under selection in the past probably, like fertility, disease resistance, we explain quite a bit less of the variance. And that, actually the square root of the variance you explain, that's where that curve is going to plateau out. We can't get above that unless we move to better DNA markers, markers that are doing a better job of actually tracking these mutations. So that's where whole genome sequencing comes in. And I'm sure I don't have to tell this group that there's just been an incredibly dramatic drop in the cost of sequencing whole genomes, where we're now at the point where we can consider sequencing bulls or individual plants for their entire genome. And that's the ultimate DNA markers. In fact, it moves away from DNA markers completely because you should have the mutations actually affecting your trait in the data. So you're not relying on a marker to track the mutation, you've actually got it. Now, I don't know what you charge for the cost of sequencing a whole genome, but in our lab it's somewhere around $3,000 to $4,000. That's much, much cheaper than it was, but it's still too expensive to contemplate doing tens of thousands of individuals that we need to estimate the effects of these genes that usually have pretty small effects. We still need really large animals. So the next challenge is to come up with a strategy where you can reduce the cost of sequencing. How are you going to do that? And in our, at least in our dairy populations, that's pretty obvious, I think. One way you can capture the genetic diversity in the population is sequence these really key ancestor bulls that have left behind many thousands of daughters in the population. And if you sequence those, you'll get the majority of chromosome segments that are floating around in the population. So these are actually 2 US bulls. This is Chief and this is Mark. And Mark in particular, he has, I think, 60,000 daughters just in Australia, something like that. Enormous numbers and huge numbers here as well. So these 2 bulls contributed an enormous amount to the population, and Chief is actually Mark's father as well. So gives you some idea about the diversity of Holstein pedigrees. But if you start sequencing these really key ancestor bulls, you're going to capture a lot of this genetic variation. So this is exactly our strategy. Our strategy is to sequence these key ancestor bulls and then our reference population, say these 10,000 cows, we can genotype them with a much lower density SNP array, say 50,000 or 800,000 markers. We can use those SNPs to track which chromosome segment they've got from their ancestors. And then we can impute the rest of the sequence on that chromosome segment. So that's the strategy that we're taking. Okay, and then you can run genomic selection on the sequence data and hopefully get an improvement in the accuracy. Okay, so I said sequencing was still reasonably expensive to do all these ancestors. In the dairy industry, I think we've worked out a quite nice collaborative approach. Because Holstein cattle are a global population and their ancestors could be in the US, they could be in Holland or wherever, we've tried to get all the groups around the world that are sequencing to sequence key ancestors but not do the same ancestor twice, and then put that into a common pool. And that's our 1,000 genomes project that we have running. So we've got 14 institutes from around the world. They're all putting their sequences of these key bulls into the same place. We're up to about run 3.0 now, and the last run we had 429 sequences of these key ancestors. And that gave us 31.8 million variants detected in the sequences of those variants. And a lot of that variation is coming between the breeds. It's like having different strains. A lot of the variation is between the strains rather than within the strains, because that's quite a lot of polymorphism. So the next challenge is to actually use those 32 million variants in our genomic predictions. And that's one thing we haven't done yet, so I can't show you results on that. That's going to be the major challenge for the coming 12 months. One thing we have done though is look at how accurately we can impute this sequence information. So I showed you our strategy for imputing the sequence information into our reference set. We have done some work to see how well we can do that. And what we have in this graph is, this was actually done by Rianne van Binsbergen at University of Wageningen. She took the sequence data set, she dropped out 20% of it, and she knocked, for those 20%, she knocked their genetic information down to just the SNP array, down to just what you would have on the SNP chip. And then she used the rest of the bulls to impute that sequence information into those with the SNP chip. And she did that 5 times in 5-fold cross-validation. And this is the accuracy of imputation that you get. This is how well what you impute agrees with what the real sequence data is. So on the x-axis here we have the minor allele frequency, and on the y-axis is that accuracy of imputation. This was a bit alarming when we did it, because you can see if you've got common variants, you do a pretty good job. You're not doing too badly. But as soon as you get a rarer variant, the accuracy of imputation really drops off. So, and we really, for a trait like fertility or disease resistance, for example, we think that a lot of the genetic variation might be down here. So another major challenge we have is to try and increase this accuracy of imputation. So that's where we're at with the real sequence data at the moment. The next step is actually to do our genomic selection on that information. We haven't done it on the real sequence data, but I'll just show you quickly something we've done on simulated sequence data, because it's quite revealing, I think. So just quickly on genomic prediction methodologies, these are methods to predict your SNP effects from your reference population. When you do that, because you've got so many more SNP effects to predict than you have reference animals or individuals, you might have 50,000 SNP effects to predict from 2,000 animals, for example. You have to make some assumptions to help you when you do that in the statistics. So one assumption you can make is that all the SNPs are going to have an effect they're all going to have a really small effect, but all of them have a small effect. So they're all tracking a gene. Another assumption you can make is that some of the SNPs have no effect. You can imagine with sequence data, 32 million SNPs, some of them just aren't tracking any QTL. So that's our Bayes-R method. So this method, top one, it gives every SNP an effect. This one actually knocks some of those SNPs out of the model and says they have no effect at all. So just to show you quickly on the results we've got from simulated data, if you just ignore this half of the graph and just look at this half of the graph, what we have is, this is in generation 0, so the same one generation away from the reference population, predicting the offspring of that reference. This is when we use the full sequence data, but we use this method that says all the SNPs are tracking some gene with an effect, tracking a QTL. That's our BLUP method. This is when we just have the high density genotypes, so the SNP array genotypes. And on the y-axis, what you have there is the difference in the accuracy. And you can see the sequence gives you no extra information at all. That's pretty disappointing when you've paid all that money to do it and run all those imputations. But we think it's an artifact of the method. We think it's because the method doesn't knock out any of the SNPs, so it has no way to distinguish between all the extra information. This other method that we call Bayes-R, this is when you apply it to the sequence data. And what it seems to actually be able to do is zero in on the mutations actually affecting the trait. So it picks those out and sets the other SNPs to 0. And that gives you a bump up, significant bump up in accuracy. It's also better just with the high density genotypes, but it really makes better use of the sequence. And it becomes really important when you try and predict 10 generations into the future, which is what we've done here. This is like running your breeding program just on the DNA marker information for 10 generations. And there the BLUP method really drops off, while this other method does well. So the message there is you have to use one of these methods that knocks out SNP effects if you're going to get advantage from the sequence data. Right, so just to finish off the talk then, I'm going to switch gears now and talk about microbiome predictions. So I don't know if any of you read The Economist, but a little while ago they came out with this edition with the title Microbes Maketh a Man, and the journalists in The Economist were getting really excited about the stories that were coming out from human genetics mainly, showing that the composition of our gut microflora was associated with many different diseases, obesity, diabetes and so on. They could actually find associations between which bacteria and abundance in the gut with these different diseases. So they wrote a whole article on that. So in humans, of course, we have our symbiotic microbes in our gut and on our skin and so on, but so do our agricultural species. If you think about plants, you've got bacteria and potentially viruses and so on around the roots and on the leaves and so on. In cattle, particularly in cattle, you've got the rumen, which is basically a huge fermentation vat of different bacteria breaking down the vegetative material. So the question we had is, can we associate the microbiome profile, so that's the relative abundance of these microbes in things like the rumen, with traits that we're interested in? And if you take that one step further, can we improve our prediction of future phenotypes using information from the microbiome? So the genomic selection works on the genetic component. The microbiome could actually explain another component. And can you use that to improve your prediction of how well that individual's going to perform in the future? So in cattle, one trait we're really interested in at the moment is methane. And methane, of course, has a very direct link to the rumen, because in the rumen you have these methanogens that are actually producing the methane. So in cattle, we were asking the question, can we use these rumen microbiome profiles to predict how much methane individual cattle will emit? One challenge for any of you that have ever dealt with microbiome data, one challenge you have with things like the rumen is the vast majority of bugs, bacteria in there are undescribed. There's no database you can go to and just say, give me all the bacteria and microbes that are in the rumen. And this will be true for many, many microbiomes that we're interested in. They just haven't been characterised. So one problem you've got is, even if you, if we go back to this cow, this thing here is actually a window into the rumen. So you can open this up, put your hand into the cow, pull out a sample of rumen, and then extract the DNA, put it on the sequencer, and get oodles of sequence out of that. If you do that, what are you actually going to do with it? So the first challenge we had was, what do we use as a reference? There is no known list of organisms for our microbiome. So what we did was assemble all the possible fragments of DNA in there. We assembled our rumen metagenome sequence coming from all these microorganisms into little fragments of DNA and called contigs. So you can imagine our rumen metagenome sequence consists of these little contigs, and it's in the order of, I think, 500,000 of these things now. So 500 little fragments. Once you have that, what you can do is start to quantitate the relative abundance of, you're actually quantitating the relative abundance of the contigs, these fragments of DNA between samples. And these are like a surrogate for the different species, I guess. So you can imagine cow 1, you pull out a sample of rumen, put it on the sequencer, sequence her rumen, and you get this profile. You do cow 2 and you get this profile. You can actually quantitate that. So here you have 3 reads, sequences mapping to this contig, 1 mapping to this, 4 mapping to this. Cow 2, she has a profile of 2, 2 and 3. This is a small example with 3 contigs. You can extend it up to our 500,000 contigs. So then you get just a matrix of sample, your cow or your plant where you took this thing from, by contig and count. The next thing we did was, inspired by our genomic selection algorithms, the algorithms we've been using for the past few years, we constructed a metagenome relationship matrix between the animals. How much of the variation was in those contigs was shared between the animals. And you can actually just multiply this matrix by itself, transposed, and you'll get the similarity between animals in their profile. Once you have that, you can use that to predict animals, as long as you have a reference population where you have phenotypes and these microbiomes characterised, you can start to predict new animals if you have their microbiome profiled. So we did this, actually it was a student of mine, Elizabeth Ross, did all the work I'm just about to show you. She tried it for 2 different data sets. She tried it for body mass index in humans, and she had 84 samples from Denmark, where she had body mass index measured as a quantitative trait, and she also had microbiome samples, sequences from these Danish samples, and that's freely available. The other one we did was methane emissions in cattle. So we had 39 cattle that were either extremely high for methane or extremely low for methane. We took rumen samples from them, extracted the DNA, and got these microbiome profiles. And then to see if we could predict anything from these, we just took another 16 animals from the herd, measured their methane, got their microbiome profiles, and looked at how well we could predict these methane emissions of these animals. And this is the answer, basically. So what you have along here is how many samples were used in the reference. So Elizabeth put more and more samples into that reference, and that increased our prediction accuracy. So this is the number of samples in the reference. This is the correlation between our prediction based just on the rumen microbiome profile or the human gut microbiome profile and the actual phenotype for that individual. So it's how well we could predict. And what you see is as you increase the number of samples, you increase the accuracy. So the blue line is for body mass index and the red line is for methane emissions. And these correlations actually surprised me. They were surprisingly high. If you tried to do genomic prediction from just this number of samples, your accuracies would be very close to zero. Okay, one quick question if you're thinking about doing this is how much money do you spend on each sample? How many reads do you need per sample? And this is just a graph of how many million sequence reads you need per sample to get good accuracies. So 3 million is where it starts to plateau off. And the good news is to get 3 million reads, that's not very expensive. That's in the order of, I think it's about $100 per sample. So it's definitely doable on a reasonable scale. Okay, that brings me to the end of the talk now. So to conclude, genomic selection has the potential to double the rate of genetic gain in some species, where we have this opportunity to reduce the generation interval. That's where it's really beneficial. It also opens up the opportunity to select for new traits that contribute directly to efficiency of food production. So feed conversion in cattle, that's pretty hard to select for usually because recording the phenotype costs an enormous amount of money. So you can't do it out in the industry. But you can do it in research herds and then translate that using the markers out to the industry. And I'm not sure, but I would guess things like water use efficiency in crops is also quite expensive to measure. The way we're heading with genomic predictions in the future is to try and incorporate this sequence data into our predictions to improve the accuracy and to improve how well they hold up over generations. And there's also this new line of research which is to use the DNA profiles of the microbiomes surrounding our agricultural species to try and improve our predictions of future performance. So this won't operate on the genetic component. This is perhaps operating on some of what we would usually throw away as the environmental component. And that might enable us to improve our predictions. Of course, there are many, many questions there, like if you take a microbiome from early in a plant's life, how well does that predict what's going to be there later in life? There are many questions like that that have to be answered. But at least I think we're potentially some way along the line of having technologies which can help us meet this challenge. So just to finish up, I'd like to thank the workers that produced a lot of the results that you saw. Elizabeth Ross, Jenny Price, Hans did all the 1,000 bull genomes crunching, which really made the computers run, and also the 1,000 Bull Genomes Consortium that we work with. Okay, I'll leave it there.